Hi everyone, this project is about taking One Health approach to evaluate the role of oil palm habitats in biodiversity conservation and spread of vector-borne and zoonosis in Malaysia. Project leader is Professor Dr. Sazali Abu Bakar, also a Director of Tropical Infectious Diseases Research and Education Centre, University Malaya, Kuala Lumpur. For Team Malaysia, other TDREC researchers that will be involved include myself, Dr. Zubaida Yaakob, a vector biologist and ecologist, Dr. Nohidayu Sahimin, parasitologist, Dr. Long Shi King, a bacteriologist, and Dr. Lau Van Loon, a molecular biologist. These four will act as a co-investigator and be the key player in the sampling activity, arthropod identification, molecular detection, bacteria culture, and data analysis. Our international partners for this project include Professor Dr. Serge Morin, a Director of Research National Centre for Scientific Research, Montpellier University, France. He currently holds several other positions, including an adjunct professor at Faculty Veterinary Technology, Kasetsart University, Bangkok, Thailand. And he also is an invited professor at Faculty Tropical Medicine, Mahidol University, Bangkok, Thailand. Professor Serge Morin will be involved in designing the project methodology and data analysis. Our next partner is Dr. Kittipon Chaiseri from Mahidol University, Thailand. He will play a role in project designing, ectoparasite taxonomy, and development of research methodology. Our third partner is Dr. Wayan Suwana from University of Mataram, which will be contribute to the entomological sampling. And last but not least is Dr. Upik Kasumawati Hadi, a professor at Bogor Agriculture University, Indonesia. She will be involved in project designing, field methodology, and sample identification of mosquitoes. This project is structured by three main objectives. First, to ecologically evaluate the diversity of animals such as rodent and arthropod vectors such as mosquito and rodent ectoparasite. Second, to detect and characterize pathogens circulating in oil palm plantation areas. And third is to engage with the communities, which taking into account gender involvement, that living within the oil palm areas through knowledge transfer programs on One Health. Methodology in brief, for the first objective, 100 rodent traps and 5 light traps will be employed across study site for 5 days, which will be checked daily. Associated environmental data will be taken at the time of each sampling. Rodent trapping, identification, and sample preservation will follow Herbitu et al. 2011. Ectoparasite will be preserved in liquid nitrogen, with some brought back alive for the arthropod cell culture activity. All data will be subjected to a network analysis. For the second objective, all identified rodent, ectoparasite, and mosquitoes will be subjected to DNA RNA extraction, PCR, and sequencing for the specific pathogen detection and characterization. For the third objective, villages located within the study area will be identified and approached for a community outreach program. This preferably school kids, teachers, and parents. The knowledge transfer workshop on vector and zoonotic in the scope of One Health will be transferred via an interactive and effective health communication. Pre and post questionnaire will be subjected to data analysis to evaluate the level of knowledge, attitude, and practice towards One Health concept, zoonotic, and vector-borne disease that's circulating in their environment. All of these useful data that will be obtained from Objective 1 to 3 will be translated into a scientific research papers. This project will be conducted at the Basir Ismail Research Complex under the University of Malaya Research Centre located in the state of Johor, Southern Peninsula, Malaysia. The basic facilities include dormitory, researcher room, kitchen, multipurpose hall, and the most interesting part is we have 400-hectare living lab to be explored. This is the milestone for this project. In brief, there will be 3 times of field sampling from March to May, detection and characterization of pathogen as well as all data analysis will be completed in September. The community outreach activity will take place in August, and this project is estimated to be finished by November 2022, which will be closed with researchers meeting to initiate new further in-depth research. 5 dissemination method, namely at least 1 scientific publication, professional project videos, mass media, policy papers, and support my own training on One Health and environment. That's all the summary for this project, and on behalf of TTRAC, I would like to say thank you for this great opportunity.